So I am happy to welcome Josh here. Like he is going to tell us loads about everything regarding to how the brain computes and with exquisite care for biological detail and experiments. So all these hypotheses we've been talking about up until now, he's going to help us unpack. And this is the best introduction I can think of.
Yeah, so I will give you a series of lectures over the course of the class. So this, I will kind of refer back to this because partially what I'm going to do is explain to you some biological, almost biological supplement to the computational models and show you how the brain actually computes things. In order to know that, you need to actually do these experiments. So everything that I will show you, there's a real experimentation behind, and so this lab visit will show you how those data are actually collected. And then you can think about, you know, every experiment has its pros and cons, so you have to somewhat also consider the realities of the experiment, the possible downsides of them. What are you missing by doing this sort of experiment versus another experiment? And I think that's why neuroscience is such a multidisciplinary and technologically diverse field, because every weak experiment kind of complements a different weak experiment, and then we can all kind of put it together into something greater than any of one of those parts. And I don't mean weak in terms of that you should doubt them, but rather there's no perfect experiment that doesn't have any downside at all. And so you have to kind of take into account all those cons and then combine together the knowledge from different methods to see what is the most true. So that's what I'll try to show you with this. So you already had the lab visit of the, and by the way, feel free to interrupt me at any point. This is mostly a discussionary session. Yeah. So you already had the lab visit for the anatomy. So you kind of see how we know things about anatomy in the brain. And actually in a future session, I will show you a little bit more technological detail of how we know things about the kind of minute and detailed connections within the brain that takes very specific techniques. But for this upcoming lab visit, you're going to see more about how we know about how neurons communicate. So in a very, very dumbed down, like simplified way of doing this, I'm going to say that to each other. So they speak to each other with their own language, which is of course in this chemical electro system where they will send, I don't know if people can see my mouse, but I'll kind of use it as a pointer. But they will send electrical signals down their processes, of which they have one, and they will contact subsequent synapses. So this is the most basic possible circuit you could have, which is just two synapses with, or one synapse with a presynaptic neuron and a postsynaptic neuron. And everything is built from this basic coupling. And you can imagine the sheer network and the extraordinary number of connections that actually take place in the brain. But this is kind of the fundamental relationship between neurons. The presynaptic neuron, which has an action potential, move down its axon, and then it contacts the postsynaptic neuron, and you have your synapse, where very specific chemicals will move across that space and activate specific channels on the postsynaptic side. So this is the way neurons speak to each other. And fortunately, because they are electrical, they're relatively easy to record using electrophysiology. But also there are many other features that that neurons exhibit that allow us to kind of listen in on their conversations and listen in on how they communicate with each other, or to look for something that is an indirect change that is indicative of them speaking to each other. All of these ways allow us to try to understand this, but fundamentally what we're looking for is this conversation between neuron A and neuron B. And we might look at that very broadly by looking at huge clusters of neurons, but this is fundamentally what we're trying to understand. So the basis of all behavior is this sort of relationship. So this is an extremely simple circuit, but as soon as I start to add in an actual behavior, you can see it gets immediately complicated. So this is the knee reflex that when you go to the doctor and they're testing your reflexes, you don't have to consciously think about this movement because it's just a subspinal circuit connection where you have your sensory neurons that are within your leg here. They send information back to neurons within the dorsal root ganglion. There's a synapse here within your spinal cord. There's actually several of them. And then they send, these are motor neurons that send synapses back to the muscle. So even in this extremely simple circuit, I've now complicated that last picture I showed you by making this 1 synapse, 2 synapse, 3 synapse, right? So this language is an immediate transduction of a sensory experience into a motor output, but it's the same basic system. So you have the neurons speaking to each other and they turn something from the real world into something within the neural language and then allow you to move and then move within the real world again. So this is the sensory motor kind of integration and transduction that we will talk about many times, but this is why we're interested in that relationship I just showed you, because that is the basis for behavior, these neurons speaking to each other. So the neurons speaking to each other, I showed you this extremely simplified version. The reality of it is much more complicated, and that's part of what the experiments that you will see at the lab visit will show. So as I said, you have this action potential that moves down the axon of the neuron. It then causes a release of chemicals at the synapse, which then signal to the next neuron. So it can also potentially have an action potential and signal onward. And that action potential looks something like this. So you have neurons that are sitting at an electrically negative position. I think you've already seen the Hodgkin-Huxley models, and you should have a understanding of what's going on here, where you have neurons that sit, they rest at a negative membrane potential, and then once a stimulus comes in, either from the world or from another neuron, or even sometimes a spontaneous event from the neuron itself, it can trigger an action potential, and that triggers changes in the sodium, potassium, calcium channels that rest on the membrane of the neuron, and it causes this sharp spike, which will go positive for just a very brief moment and then negative again. And this whole activity here, the action potential, can take place in the matter of milliseconds, but it is the basis for how neurons speak. So that's what's happening down the axon. And then at the synapse is this sort of connection where you have this little axon bouton and you have vesicles, extremely complicated, even more complicated than this schematic, that will release chemicals that signal to receptors on the postsynaptic site. So this language is the basis of how they speak to each other. These electrical movements of ions that are moving in and out of the neuron at all times, and then very specific chemical release at the synapse. So neurons are speaking to each other in this way, and they are doing that within the context of an unbelievable complication. So the music here is not really necessary. I'm going to mute it. So this is just one little section of the mouse brain that someone has now made into a 3D reconstruction of the existence of these neurons. And we're just going to move in, and you can see how packed full the brain actually is of neurons. So each one of these little balls here is the soma of a neuron, and the line coming up above it is its dendrite. Below it will be the axon, and all of these are interconnected and organized in very specific ways and talking to very specific other neurons in order for information to pass through the brain and for behavior to actually take place. So that very simplified structure that I showed you, I already kind of further complicated by explaining the electrical component and the chemical component. And now on top of that, you layer the extreme density of the brain and the number of neurons and the number of connections that are taking place. So all of this has to be kind of taken into account as we try to measure neural activity and actually see how the brain is working. So I'm going to pause that. And of course, we typically do not record from the human. We can, but not in the methodologies that you will see next week. We're typically recording from the mouse brain. Sometimes this can be done in a monkey brain, which is not done here, but is done elsewhere in the world. The mouse brain, although it's obviously much smaller than the human, is quite analogous. And so we can find analogous structures within the mouse brain that match the human brain, and it allows us to understand a lot about brains in general. Even the fly brain or the brain of the worm can be extremely informative for how the brain works in general and how the human brains works, and we can extrapolate from that information. So this will bring us to the actual techniques that we use in measuring neural activity. And I wonder if anyone has any suggestions for how you might measure neural activity, given all I've told you so far. How sometimes it's there's this very simple relationship that we want to understand, but then we have to take into account the complexity and the context that those neurons live in. And I wonder if anyone has any suggestions for neural activity measurement techniques. Any ideas? You can just talk if you have one. Or feel free to drop it in the chat if you prefer that. But if you unmute and have a suggestion for a technique, go for it. Yeah, Maxim. I can't hear you actually, Maxim. Oh, wait. Oh, Mario, you're muted. That's why I can't hear you.
Yeah, I said it probably some microphone levels from Maxime, because I can see he's unmuted, but
yeah, I know.
Hear you.
Yeah, that's what's confusing. Now you're muted again, though, Maxim.
Yeah, went in and out. You continue. Unfortunately, you're not coming across.
Yeah, I don't know why. You might want to draw. If you have a suggestion, feel free to put it in the chat, though, and I'll read it out.
So people, Isa
yeah,
has been suggesting EEG, and now she's basically sent a message directly to me first, and now to everybody in the chat. And single cell recording.
EEG is a very good one. Maxim, what do you mean by single cell recording? Is it possible to be more specific, or do you want me to just discuss that as a general topic? Yeah, so measuring the activity of the neuron. So that's still quite broad, but I'm gonna explain some different techniques for how you would do that. So part of what I was hinting at before was that we have these ways of kind of eavesdropping on neurons, and neurons speak in a very particular way, which is that you have this ion movement in and out and this electrical gradient, but that type of way actually lends itself to eavesdropping in a few different ways. And so I'll kind of show you some options on that. So single cell recordings, I will show you at the lab visit, and you will see in a couple other contexts, but often you'll see things at a multicellular way. So you will actually benefit next week from very wide array of techniques. And so you'll see a few of these. EEG, unfortunately, you will not see, but you will see things that are similar to EEG in terms of their technique and in terms of their kind of scope. So I think these are very good suggestions, because they kind of remind people of the different levels of measurement that can take place. And so these are some of the different measurements of electrical activity or neural activity that you can use. And I say level because it kind of refers to the resolution that you can detect and the number of cells, and actually even the pieces of the cell that you can record from. So EEG kind of exists up here, and that's when you're measuring actually even through the scalp. You're not measuring directly on the brain or directly in the brain. You're measuring outside of the brain in a completely non-invasive way. Which is really handy for measuring the human brain. It's also handy for measuring from animals when you would prefer to be non-invasive, or you can be non-invasive, but the resolution here is really low. So, and by that I mean the spatial estimation of actually where the signal is coming from, that resolution is low because you are collecting from so many neurons at once. And obviously you can see the sulci and gyri of the brain, and it makes it difficult to pinpoint. So for instance, if you're recording EEG, which is obviously electrical recording from here, you don't really know if the signal is coming from inside this gyrus or from the top of the sulcus, etc. And that leads to the resolution. Yeah, Maxim also mentions time resolution. So time resolution is really an interesting feature. EEG has extremely high temporal resolution. So it can pick up events at really fractions of a millisecond, but you're then picking them up really broadly across the whole brain. But EEG is often used for sensory detection or timing of a movement in correlation with the brain. So the temporal resolution is really high, but you're right, it's the spatial resolution that I'm referring to. You can have low temporal resolution as well in this circumstance with something like fMRI, which is quite a slow process. And so you won't see fMRI at the lab visit, but you will see something that uses a similar concept, which will be fast ultrasound, actually, which is looking for also blood flow dynamics, which are not a direct measurement of neural activity, but they are an indirect measurement, because if a bunch of neurons are active, they need more oxygen and more blood flow will be delivered to that area. And you can actually then make assumptions about activity that's ongoing at the time. And so the ultrasound and the fMRI are going to use a very similar reasoning, even though the technique is very different. And their resolution is lower, but the fast ultrasound is actually not that bad in terms of its resolution. So I'll show you an example of that. The EEG could be then, instead of being on the scalp, it could be moved to the actual surface of the brain. This is often done during surgery. If the neurosurgeon wants to make sure that they're not going to remove an area that's very necessary for language or very necessary for fine movement, they can place an ECOG directly on the brain and record. You could also record from extracellular multi-electrode arrays, where you have this multi-electrode array here, and you can actually grow neurons directly on top of that. You won't see that live, but I will show you there. I'll show you here in a moment an example of that. And you could have intracellular recordings where you actually put the electrode directly in the neuron to record what's happening from the neuron's perspective. So I will show you a bit of that at the lab visit. And what you will not see is the really fine detailed channel recordings, which is a subfield of neuroscience all in itself, which is actually looking at dynamics of single channels on the neuron. It's fascinating work. But that's just not the sort of field that the people in this department are in. There's not a lot of biophysicists really here, but this is a fascinating field in and of itself, but we will stop around here. So you should be able to see a lot of the stuff in these levels at the time. So not necessarily EEG or fMRI, but a mouse equivalent of that with the fast ultrasound or the functional ultrasound. And then you'll see a lot in this area. The difference between EEG and MEG, oh, actually, I'm blanking on MEG and FNIRS, actually. I'll have to come back to you on that to make sure that I don't say anything incorrect here. But the MEG is likely just a different location, but I can check on that. So I'm going to show you kind of a concept that you won't see at the lab visit, but I think is good to introduce, which is the idea of studying an isolated neuron. And so for an isolated neuron, you could either grow it outside or remove it from the brain in order to study it's outside of its typical context. This allows the researcher to study the neuron without the influence of the rest of the circuit. So I showed you that really complicated circuitry, but sometimes you want to isolate something so you can reduce the number of variables that you're recording from at the same time. So for instance, you might use an organotypic slice. So that's when you take, you dissect the brain out of, for instance, here, a mouse, and you keep the brain alive using special mediums. So you can actually slice the brain into thick slices of, for instance, here in the cerebellum, and you can then drop the cerebellum into a dish. You keep it alive with a certain culturing medium, and the neurons will still be active and will still grow and migrate. And you can actually see an example here of, oh no, okay, I'll have to send you this video later. I didn't update it, unfortunately. But here, this is actually an organotypic slice that was a really beautiful example of a neuron migrating down a glial process. And you can actually use that in, you can actually monitor that in the slice with real-time microscopy and video, and keep the slice alive for quite some time. Yeah, in this case, you can watch a neuron beautifully migrate down this process. So I'll find this video and upload it later on. So another option, which is done in this department, but you unfortunately won't see at the lab visit, is to take the neuron, dissect the brain, and then actually mix the neurons up into kind of suspension. So they're completely outside the context of their normal structure. And then you can grow them onto a dish. And in that case, you end up with neurons that have their own connectivity and their own circuitry now in a dish, outside of the normal context of the brain. And so in that way, you get to study the neurons in isolation. You get to study things like the growth of their processes, like their axons and their dendrites. That can be easily studied in a dish. And they will form synapses with each other, and you can actually monitor synapse formation and record from neurons during that process. And you can grow them on these multi-electrode arrays. And so that's a nice example of what I was showing you before. These are now going to be extracellular recordings where the electrodes are just around the neurons and the neurons are just growing right on top of them, and you can monitor them the entire time. And so a culture, as it's growing and developing, can be recorded, and you can track the electrical activity, and you can track it on a grid, so you know where in the culture this thing is actually happening. So if we want to record from other neurons, and I apologize if this is a sudden image, so this is actually a dissection of the brain, which you will not see at the lab visit. You will see only kind of the setup and the theory behind it, but this is essentially how the brain has to be harvested. So in order to study these things, we do have to sacrifice mice periodically and harvest the brain and actually slice the brain in order to record from neurons. And this is done with great care and with great speed. So in order for the brain to stay alive, you actually have to learn how to dissect the brain out in less than a minute. And then you immediately get it into solutions that will mimic the fluids that are endogenous to the brain and continue providing nutrition and support to the brain so that the neurons will stay alive and you can record from them. In this case, this person is planning to record from somewhere in the frontal brain, and so they're going to slice here and place it on a platform and literally glue it down, and then we have a special slicing machine that will cut it into very specific 250 to 300 micrometer slices like this, and then we will keep the slices alive all day long and record from the neurons one at a time. And so this is a case of acute brain slices that allow you to record from individual neurons, single neural recordings in this case. So this is similar to a setup that I will show you next week. This is the slicing equipment. I will go through it with you, but just so you kind of are aware of the pieces of it, you need to have the actual place where the brain sits and the place where the electrode can then interact with the brain. So this is a slanted electrode that can be controlled very finely with this micro manipulator. And so you can actually control and make movements of less than a micrometer at a time. And you can bring the electrode very closely to the neuron You're not blind in this circumstance. You have a microscope and a camera that can then show you where you are in relationship to the neuron. And you have to actually get the electrode right up to the neuron. So the electrode has to be smaller than a neuron, and then you can break into, quote unquote, the neuron and actually make your electrode one with the neuron and record from the perspective of the neuron. The micromanipulator is a piece of equipment here that will control this electrode, and you can move it in all three directions. So what you see here is three different wheels that you can actually turn manually, but it converts your manual movement into extremely tiny movements of the electrode in all three dimensions. So you can move it forward, back, side to side, up and down. And you can move it in very, very tiny ways because you need to be able to go very close to the neuron and not damage it. So it requires very, very small, careful movements. And so this is the micromanipulator that allows you to make micrometer level movements. Yes, so the solution in the dish is indeed comparable to the CSF. And so actually for this recording, all day long, you're recycling and flowing through something called ACSF, which is artificial cerebrospinal fluid. So we know well enough the compositions of the CSF in order to replicate it, and that's how we keep the brain alive. So we and there's a limit to how long you can keep a brain alive outside. There's a limit to how well these artificial solutions can maintain the normal conditions, because obviously the best condition is still in the head and in the body. But we do have solutions that allow you to keep the brain alive well enough that you can record all day long, or you can actually maintain something in the dish. So I'm showing you this one in more detail because this will kind of show you the general concept of what needs to happen here, right? So you have the brain in whatever condition your technique is optimal for. You need something to interact with it in order to monitor the activity, and then you need some way of recording it. And so in this case, you have the computer to record it. You have the electrode to interact with the neuron. So you need to physically interact with the neuron in some way. And then you need to be able to actually have the brain or the tissue available for you. So those are kind of the three key elements of this. And those will remain there, but they will change dramatically depending on the exact technique that you're using. So here again is kind of a schematized version of this. You have your recording chamber where the brain tissue sits. You have your electrode holder holding your electrode. And then you have the ability to see it using the microscope objective, the camera, the computer screen. And then, of course, the electrical activity is brought to a whole system of amplifiers and digital converters that can then be recorded by the computer. And so you have to translate the neural activity into something the computer can read and record. In this case, it is the electrical activity of the neuron, because this is an electrophysiological activity. In a different case, we might record, obviously, blood flow, or we might record fluorescence if we're recording in a calcium imaging experiment. But the basic setup is the same. You need a way of interacting with the brain. You need a way of monitoring the brain and converting that system, that information, into a system that can then record and be digitized. So another example of the sort of thing. Oh, sorry, I missed a question. Yeah, the pump, indeed. This is for providing the ACSF all day long to the brain tissue. So you pump it through and you actually carbogenate it in order to keep the pH balance of the ACSF all day long continuous, because the brain will be taking from the ACSF and you have to replenish it throughout the day. And so that is indeed what the pump is for. And that's not going to be necessary for every experiment. Some experiments, if they're in vivo, obviously the CSF is being provided by the body, so you don't need it provided in that case. But for acute experiments like this, where you fully removed something from the brain, you then need to provide something that will sustain the tissue and provide nutrition for it. So here's, for example, the type of thing that you might see in a slice experiment. So in this case, these are some terminologies, and you'll hear jargon like this for each technique. So you have to be somewhat flexible in your thinking. When you hear jargon, note that you don't know what that means. And then at some point you can ask, can you define that term? Or you can interrupt whenever you're at the lab visit and make sure that you understand. So in this case, I might use jargon like the bath. And the bath in this case is just the ACSF fluid that's sitting around and above the neural tissue. So in this case, what I will see is I will have an electrode, which consists of a glass sharp pipette and a wire inside of the glass with an internal solution that will mimic the internal solution of a neuron. And you have the bath, which is just essentially a puddle of ACSF that is provided and helps to keep the tissue alive. In this case, you will have the pipette approaching the neuron. You then can provide little electrical pulses and read the voltage and the resistance of the neuron. So here you have current and you have voltage. So if I provide little current pulses, or if I provide little voltage pulses here with the neuron, I can then monitor how much current changes at the level of the electrode in that case. And so in this case, when I'm just in the bath, the electrode is just far away from the neuron, just in the bath, I provide voltage steps, and I have a free flow of current that essentially matches that voltage step. As I get closer to the neuron and I press up against the membrane, what I'm looking for is a tight seal on the membrane so that I can really make a strong connection with the neuron. And in this case, I provide the voltage step, and the current flow will be tiny because nothing can now move. The membrane is now pressing against my electrode, my glass electrode, and I now have something called a gigaseal. And so here's another example of some jargon that I will use for this certain technique. And in this case, nothing can escape this connection. So I'm now sealed up very, very, very strongly with the neuron. And the next step is to actually break the neuron. And I'll show you an example of how you might do this. And you literally do this with your mouth, with a little mouth pipette, and you go pop, and you actually break the membrane inside of the glass pipette with the neuron, and you start to now see a response that looks like this. So the voltage signal that I was giving the whole time doesn't change, but the response is wildly different. So once you break into the cell and you become whole cell, quote unquote, then the same signal will produce something like this, and that you will see a large change. And then what you see here is a capacitance charging curve. And I'll explain in more detail of what that actually indicates and tells you about what you're recording at the lab visit. But I'm just giving you examples of the sorts of jargon that you might hear, and you kind of have to wade through them a little bit and pick them out when it's really unclear what it means, and get to the meat of the matter here. So what this means is that I now am continuous with the neuron. My electrode and my neuron are now one thing, and I'm recording from this perspective of the neuron, keeping in mind that the neuron's membrane is a big capacitor, and that's why you see this capacitance charging curve whenever I give a voltage pulse now, right? Let me check. Oh yeah, that's been Mario. Yeah, so the ACSF and the CSF, I will explain in more detail at the lab visit. Because that's a big part of how you keep the neurons alive. But essentially, the CSF is a cerebrospinal fluid, which is the fluid that's flowing all around your central nervous system at all times. And it provides nutrition to your brain and your spinal cord and neurons. And it is what we're mimicking with the artificial cerebrospinal fluid in order to keep the tissue alive and record from living neurons, even though we've removed them from the context of the brain. I'm going to skip this for now. These are just different types of recordings you can make, but I'll show you actually examples at the lab visit. But so this is actually a sort of thing you might see where you see a slice of brain tissue, you see a recording electrode, and you can actually see stimulating electrodes. And so part of the benefit of a slice recording is that you can now induce plasticity events. So you can record from a single neuron, you can then stimulate synapses onto that one neuron, and you can actually induce what we call LTP, or long-term potentiation, which is in theory the sort of mechanism that allows for learning and memory in the brain. That's how synapses change. That's how connections become stronger. If you have a sudden, very important event and your neurons really need to understand, oh, this was significant, I need to keep this connection very strong, this is the sort of mechanism that might underlie that sort of learning and memory. And so this is a way in a very artificial setting to test for the mechanisms of learning and memory. And so I'll show you a little bit more detail about that, but that's the sort of benefit, even with all the cons of removing tissue from the brain and the context of the whole brain, it allows you the pro of being able to actually record these sorts of events. Okay, so now I'm going to go more quickly through other sorts of techniques that you'll see. So you'll also see an in vivo electrophysiology experiment where you actually see neural activity in the context of the whole brain. Yeah, the action potential here. So this is not necessarily an action potential, but it's rather an extracellular postsynaptic potential caused by a stimulation. So you have your recorded neuron, which in this case will be your postsynaptic neuron, and you have your stimulating neuron stimulating the presynaptic neurons. And whenever you stimulate the presynaptic neuron, you cause a response in your postsynaptic recorded neuron. And in this case, what they're showing you is that that response stays the same. And then when you give a strong tetanized input, which is just a high frequency input of the presynaptic neurons, really rapid and just actually for a short period of time, you then see an increase in the strength of that response. So now the strength of that synapse is weighted more highly, and you actually can turn the power of that synapse up, even though it's the same connection, the same sensory input or the same internal input, now evokes a stronger response. And so that's what I mean with the kind of mechanism of learning and memory that you can record in the slice condition. So the slice condition is cleaner, right? You're reducing the number of synaptic events that can happen, and you can actually flow onto the brain synaptic blockers. And so you can say, I only want to see glutamatergic activity, for instance, which is one type of synaptic chemical. I don't want to see inhibitory currents. So you can block all of that within the slice. In vivo, however, you have everything. You have all of the information going on, and especially in an awake animal. So here you have a head-fixed awake mouse with a recording chamber that gives you access to the brain, and you can put an electrode directly into the brain. We all have to thank the universe or thank whatever allowed the way things are, that the brain does not feel pain. Everything would be so much more complicated if the brain had pain sensors, but the brain does not feel pain. And so that allows us to record from the brain in awake animals and awake humans without causing any discomfort. So in this case, the mouse can be trained to sit on the wheel, and this is basically they're here for a temporary period of time, and allows for them to run freely on this wheel, even though they're head fixed, and it keeps the head fixed for the electrode to be inside of the brain. And in this case, you can record for a very long time in this case, which is indicative that the brain is actually not damaged very much in this case. So all of these things require the sacrifice of an animal, and so that's why they have to be taken very seriously. We can't be doing frivolous experiments, and this all has to go through strict ethics approval. But it allows us to understand better how the brain works, and so that's what you'll see next week. And then as we go through this class, and I show you examples from the textbooks, you have to understand that all of this came from these sorts of experiments. So all of this knowledge came from experiments like this. So in this case, we can record from a single neuron like this. So these are the action potentials of a single neuron. Each line here is an action potential. And this is within the context of the whole brain. So nothing has been disrupted, nothing has been removed in this case, and it's within the context of the mouse still receiving sensory information and moving the whole time. So you're getting all of that information together. And so it's much messier than the slice in vitro situation, but it does provide you more context for the neuron. So there's a pro and a con to that as well. So that's the electrophysiology. Oh yeah, so in this case, this just happens to be the background strain of this mouse. We have many different strains of mice. There are black mice, brown mice, white mice, and this just happens to be the strain that was used in this case. It's a little complicated of why we picked certain strains, but there are differences between strains in the mice, and so that's why whenever you're doing an experiment and you genetically manipulate a mouse, for instance, you want to have, this gets into the weeds very quickly, but say, for instance, you're curious about the consequences of one gene in the brain. So you have a gene that causes a disease, for instance. And so if that gene is manipulated or mutated, it leads to a disease in humans, and you can model that in the mouse. Because we have the way that we can breed mice is we know that you will have one mouse that has the gene that leads to the disease, and in the same litter, you could have a mouse that has that does not have that gene, but everything else is basically the same in that circumstance. And so you have these litter mate controls in this case, where you can have siblings from the same litter, where you have one mouse that has the gene of interest and one mouse that doesn't as your control mouse. And so in this circumstance, and I don't know which one this is, of course, this could be the experimental mouse or the control mouse, and they just have to be from the same strain and match to each other, so you can make strong conclusions about the results of that gene, given that you don't see that sort of activity or that sort of behavior in a mouse of the exact same litter, only with that one gene not changed. So that's probably more detail than we need to get into in this circumstance, but that's why, for instance, you might see the brown mouse here, because that is the strain that gene is kept on, if that makes sense. Okay, so that's another electrophysiology technique. You can also in vivo record neural activity, not with electrophysiology, but you can eavesdrop on the calcium dynamics of the cell. So what I showed you before was that the language of neurons is electrical, and that is the movement of ions in and out of a neuron that changes the membrane voltage of the neuron itself. And we can actually use something like calcium, which I'll go into more detail on in the future, and actually at the lab visit, you'll hear more detail about this. But calcium is one of those ions that is really closely tied to the activity of a neuron. So when a neuron fires an action potential, the neuron will suddenly have a peak of calcium ions within it. And we have really fascinating technologies that allow us to monitor calcium levels within the neuron with these genetically encoded fluorescent proteins that will bind to calcium and then give a fluorescent light signal when calcium is high. So we can actually monitor the state of calcium in the neuron based on how bright literally a neuron is. And in this case, what you do is you will head fix a mouse and you will open a window over the brain, and then you can monitor using a microscope the calcium dynamics. And what you see here are each line here represents a different neuron. So 2, 22, up to 88. You can monitor tons of neurons at once with this technique. That's why it's very powerful. And you can see here these little blips in the recording, and this indicates a calcium peak. And you can then temporarily correlate those calcium peaks with some behavior or some stimulus that you gave the mouse, or a movement, or whatever it is that you're interested in. You can actually monitor all of the different neurons in a field of view and monitor their activity correlated in time. So back to pros and cons. I wonder, I don't know if anyone can predict it. This might be too complex, but if you can predict what the con of calcium imaging is, please feel free to jump in. But just know that you record this activity using video. So does anyone have an idea of what a con of calcium imaging might be, given that knowledge? You can
Maybe that it
unmute.
costs data.
Say it one more time.
Maybe that it costs a lot of data to actually record this, and you can't record it for a long time.
That's a good point. So that's not the specific downside that I'm referring to, but that's a really important point. These are immense amounts of data, and managing these data are really complicated. And that's why a lot of neuroscientists have become de facto programmers as a result of these sorts of techniques and the amount of data that we're collecting now. But there's actually one other con that I'm specifically referring to in terms of the fact that we're not recording electrical activity anymore, but we're now recording images with a video camera. Does anyone have an idea of the sort of downside that might introduce? I don't know if I'm being obvious enough with this question, Mario, but it is complicated. So if you haven't thought carefully about this, maybe it doesn't immediately come to mind.
Maybe I can do this to help him.
Yeah. Oh yeah, that's a good point.
The video isn't like really reliable always. It also depends on like if you go really far into one neuron, you get like pixels and like the Mario shaking his video, so you cannot really see his face anymore.
Yeah, so exactly. So you have to now deal with the physicality of a video camera. So you have movement, you have the fact that you will see somewhere in a different Z plane. So here what you see is that these neurons are not all in the same Z plane. So this is the X and Y view from above, right, as you're looking at the brain, but there's actually slightly different Z planes of the brain. The neurons are not all perfectly laid out in a row for us. And there will be different focus points as a result. So obviously we focus by looking at a Z plane. A lot of these components of vision we will get into that you may not have thought about before. The way you see something is by looking physically at a plane of Z. And that's the way that we also image with a microscope. And so if you have things at a different Z plane, that can be complicated. And then the last con of the video camera is that you're taking frames. A camera is not a continuous thing. You're taking discrete pictures and then putting them together to make a video. And the rate at which you collect those frames is your temporal resolution. And so you do not have anywhere near the same temporal resolution. I can collect this information at 10,000 bytes per second or something like that. I can have extreme temporal resolution with electrophysiology, way more than you could possibly need. But with calcium imaging, you're probably imaging at like 30 to 100 frames per second, something like that, a much, much lower temporal resolution. And so in order to get valuable information, you have to repeat trials over and over again. And that way you can actually be sure that the temporal resolution that you see is reliable. You have to average trials on top of each other in order to really understand. So that's one downside of calcium imaging. The upside is huge. You can see huge numbers of neurons at once, all of their activity together, their dynamics between each other. It's a beautiful technique, but that is that the temporal resolution is a downside. So you'll see an example of that. And then last for today, I will show you an example of FUS. So this is the fast ultrasound. And so similar to, excuse me, so similar to fMRI, we're now imaging blood flow. So ultrasound, if you know an echo, if someone is pregnant, you can use an echo to actually see, quote unquote, the baby in the womb of the pregnant mother. And you're doing that by actually giving sound frequencies and then measuring the sound that is returned. So in fMRI, for instance, you give magnetic changes, and then you can actually record the magnetic resonance that comes back from the brain when you make these magnetic changes of the water molecules within the blood that's inside the brain. In this case, you're actually sending sound information into the brain, and you're recording back the sound response. And this actually can monitor movement as fine-grained as blood movement inside the brain, and that will tell you something about activity changes in the brain in relation to a behavior. So I can stop there. Oh, what is funny, Selena? Is this all sounding crazy? Oh,
No,
Mario's
sorry,
coming.
Josh.
I see.
I've actually I told him a joke as you were talking about the amount of data. I told him a joke in the chat. I'm sorry.
Oh, I see. No, that's okay. That's okay.
And but
yeah,
this allows me to ask you. So now it's 15 minutes in.
So
we can either go to the end or like a pause, depending how many more things you had in store for
no.
them.
So this is my last slide. So this is this is the end of it. So I figure now we can take a break and come back and discuss, or we can have just a brief discussion of anything that I've said that is really unclear, that you want to know more information about. But in this way, I've essentially run through the different techniques that you will see next week, so that you're not kind of caught off guard as you walk into the room. But feel free, of course, to ask me any questions, or we can wrap up the session. It's I'll leave it up to the students. But Mario, if you think there's anything I should go in more detail about,
no, I
I'm
happy
happy
to
do so.
In fact, I was thinking about how nice that. So it's a very cough-inducing, because in the end you did speak during an hour in the block, but I think that it's amazing information. So and I'm going to make sure that everybody watches this before the next lab visit with an assignment, and potentially I'm going to even ask a question about people asking questions about what they're going to see in the lab visit. I mean, to basically prep us so that we can, when we're giving them the lab visit, address some of the questions they might have. I think that's great material for review. But I also think that right now, if you guys are thinking about, let's have a little bit of a conversation on these experimental methods. I think that we have like many different windows into brain function, but we haven't talked all that much about what are the questions that are enabled by these types of, clearly you can see neurons firing, but there's also the questions regarding brain function and brain computation that we're trying to unpack by looking at this through these lenses. So I thought, let's open it up for questions and see what people, are curious about. And no matter what direction it is, Maxime at one point did say, "Poor mouse," when he has had fixed. And I think,
exactly. And I think that's not an unreasonable kind of gut reaction to it, but it is taken into account. And I will say, I always tell my students that an uncomfortable mouse is not a mouse that you want to measure from. We want to record from mice that are as comfortable and as in their normal situation as possible, given the restraints of needing to access the brain or the complication of head fixing an animal, for instance. But for that animal, by the way, that the surgery in order to implant those things on the head will take place. They will be given days of recovery time from that surgery. And then before any experiments, they will be acclimated to that setup so that they're very comfortable being placed in the setup, and they don't need to be anesthetized as they're placed in there or anything. They can just be placed, and they will learn that this is not a big deal. This is not going to hurt me, and they will sit calmly in the setup in that condition. So it's all taken into consideration, because a stressed animal is not, that's not a normal animal anymore. So it is part of the consideration for these experiments. And then one more thing, for the lab visit, you will not see every station. You will miss one. And the only way we'll learn about that station is from your fellow students at the debriefing. So be aware that you may have to explain one of these stations to one of your fellow students that did not see it. And now you won't know which one yet, but we will call on you at some point in the debriefing to do something like that. So I would recommend bringing a notebook. and keeping notes of these stations, and asking questions to make sure that you can explain it to somebody, because that's the only way they will know about it. And of course, we will discuss it and correct if there's any errors, but I think it should all be easily explained with the people presenting to you. But anyway, any questions about what you saw today?
I have a question about the ultrasound. So the last slide, doesn't the skull give a problem with making the ultrasound, or do you do it below the skull?
So in this circumstance, I believe for the surgery, the skull is removed and a window is placed instead, and that overcomes that. So so the top of the skull can be removed and then replaced with the type of glass that will replace the skull and then provide what the pressure and the support that the skull usually provides, but with a see-through instance. Yeah, indeed.
Yeah, a see-through sound-wise, right? So transparent to sound. There's one thing that I wanted to mention. Probably if Peter Krausinger was here, he would remind us that in principle, just if you know the shape of the skull, then you can also remove the skull with an inverse filter if you model the scattering within the skull. So the signal reduces power, but in principle, you should be able to say unscramble the signal that gets through the skull through ultrasound. So this is one of the grails of functional ultrasound analysis, is when they will be able to mathematically and physically model the skull so that they can then record it from outside.
Yeah, which would be cool.
Yeah,
even less invasive in that case.
Probably happening in the future. Yeah.
Yeah. Oh yeah. I mean, it's all of these technologies that I'm showing you. Some of them are really, really old, like the electrical recording, electrophysiology. But even then, the technology for those is incredible now. Like you can, I mean, I could teach any one of you to do these sorts of recordings at this point. They don't require like vast knowledge of electrical signaling. You don't have to build your own amplifiers anymore. All of this stuff we have now at our disposal, and it's just often a matter of practice. So the technology for neuroscience is increasing like crazy, and so calcium imaging, and next up will be voltage imaging once they lock down that, and that will be a game changer, and I think it will be fascinating to see the number of neurons you can record in that circumstance. But yeah,
indeed.
Any other? Who have I not heard from today? Who should I bother? Maybe
can I also
Rihanna?
ask something?
Can I bother you? Oh yeah, Maxim, go ahead. Oh yeah, we hear you, Maxim.
Yeah, finally. So can you maybe, because I didn't get it, so explain one more time the underlying principle of fuzz, like the last technique?
Sure. Well, actually, well, Mario, I don't know if you want to jump in as well, but
so do you know what Doppler signal is, Maxime?
Something with radiology or something?
No, so Doppler's interference of sound with itself.
Okay.
So, but you know anything about how people image babies in uterus?
Yeah, it's like they go there to that thing and trying
Yeah,
to see
and that thing.
It's
So that's the same thing, basically.
Okay.
So the thing that they use there. So we have clinical technologists here, don't we? So who of you, Rihanna, I think you were a clinical tech, right? Rihanna?
Yeah, and Eva is as well.
Rihanna and Eva, precisely. So
Anissa.
you
So,
know how
all three of
ultrasound
us.
works. Maybe you can give us, maybe you can give Maxime a little bit of a primer.
yeah.
Girl.
Do you have much experience with the ultrasound technology, Rihanna?
Well, it's the one we covered the least. So yeah, some basic knowledge, but I'm not sure I'm the one to explain all about it.
That's that's okay. But I mean the
So basically, the sound it echoes inside, right?
Yeah.
And what you measure is the echo of the sound you sent. So you send some sound, you get some sound back, and you measure that echo and you compare the difference between the two. You can make an image of that. And in the case of functional ultrasound, you're looking at the deformation of the arteries and veins in the brain. And you can then think that, okay, so say you are throbbing as you're an artery, you're pumping blood. From 1 millisecond to another, the way in which the sound is going to reflect changes. And that you can localize inside the brain. That's basically the image of the fuzz. Would that serve you, Maxine? I mean, is that sufficient level? Because if you want to go deeper, you would have to understand how sound waves interfere with each other and how they're measured, with that thing that you put in the belly of the lady. Or
But I
in the
mean,
brain,
this is essentially the exact
exactly.
same thing you put on the belly.
It is exactly the same thing. And you produce then two-dimensional images because your information scatters and returns into a single measurement point.
Yeah. But I will make a point that the concept of like sending something and then getting the signal back, that is throughout all of these experiments. So for instance, in the slice physiology, if we want to record the neural response to a stimulus, you have your recording electrode, but you also stimulate through that recording electrode. So you can pass a current or a voltage down the recording electrode. You then know how much you gave, and then you can record the response of the neuron right back again. So you can kind of subtract what you gave and then see what the response of the neuron is. But also for instance, the calcium imaging, this is a fluorescent signal of a green fluorescent protein, which means that it's a fluorescent light. So it's not a light that just you will see
glows by itself.
White lights. Yeah. So you have to actually send a certain wavelength of light into the tissue and then record what you get back from the tissue. You will get back a lower power light signal back. So you will send something like a blue light, and then you'll read back a green light, quote unquote. You will see a lower wavelength light come back again. Or sorry, a longer wavelength light come back again, a lower frequency. So a lot of the time, this is the case. We have to kind of send something and monitor what we get back. So with the ultrasound, you send a sound into the tissue and you monitor what echoes back to you. In this case, this is black and white because, in all reality, you
measure an intensity, which is a gray value.
Exactly. So you're measuring one quote unquote light spectrum of the green fluorescent protein. But I mean, there's no real value. You're just measuring intensity differences. So you have this, like, you can shine a light onto something and you will fluoresce back to you.
We could display it in green if we really liked, right?
Exactly. Yeah, exactly.
But it would be the same as coloring a nebulae in outer space, right? It's pseudo coloring to give us an understanding of the signal.
Yeah. So yeah, that's kind of a common feature in neuroscience where you have to send a signal and monitor.
It's true. I mean, like I was thinking about that this first time that it's very general, even in the patch clamp, right? So you do with the reference signal, everything. I mean, whenever we have a voltage or current clamp, I mean, all of them. I mean, you're sending something and then you're measuring what comes back. Exactly. This is the way we find about. It's a very nice insight. Thank you for that.
Yeah. So then fluorescence, I think, is often a thing that it doesn't really come naturally to people. It's a very strange concept to think about, but these are light signals that we can't physically see. So you need special microscopes and special detectors that can actually pick up.
You remember, I think that maybe they will remember if were ever children and they like put those fluorescent little stickers on the ceiling with stars and flying pigs. I mean, so these ones you have to illuminate so that they shine.
Yeah,
that's
or like a black light, a UV light like that can pick up fluorescence signal. So if you've ever done that scary,
right?
Thing, exactly. These are fluorescent materials.
You go to the techno rave, as we used to call it.
Your teeth are glowing then.
And then you shine blue light. You don't see the blue light shining from the lamp, but you see shining from people's teeth or t-shirts. So fluorescence,
which is technically always capable of happening. So that fluorescence element of the teeth or of anything that is fluorescent is a constant intrinsic property of that thing. But then in order for us to physically see it, we have to be in the right circumstances. So we have to make it possible for us to see it. So that's what we do, for instance, with calcium imaging. The green fluorescent protein is always there, and it's always potentially fluorescent, or in this case, changing with calcium. But in order for us to detect it, we have to put the right microscope and shine the right light on it, and then record what comes back to us again. So that's an important concept too, which is to optimize the collection conditions for the type of data that you're trying to get. I think that's a common feature as well. I learned when I was in South Africa a little while back, and I went to on a safari, and I learned that scorpions are fluorescent. Did you know that?
No, I did not know that.
Yeah, so you can shine a UV light at a scorpion and they will glow. It's incredible. You should look that up. It's really really wild.
That is, I have to. What's the?
I have no idea. I don't know why they're fluorescent, but anyway. But if you want to see a really interesting case, because in that case the scorpion's technically always fluorescing, but in order for us to see it properly, you have to hit it with the right fluorescent light.
For sure, a bee can
glow.
spot a scorpion from a very large, for very long distances, right?
Yeah,
Bee
exactly.
also sees in ultraviolet.
So I learned this because you could buy scorpion flashlights, which were fluorescent flashlights. And then there was a tree near our campsite that was just covered in scorpions. And so you shine this light and suddenly you see all these glowing scorpions on this. It was crazy. But anyway, that's to say that you need the right method in order to actually detect certain signals, right?
Yeah, I always
or else
like
you wouldn't know.
Yeah, I like the comparison with lenses, right? So we need the right everything, the right magnification, the right filter, the right. We need to know which scale we're looking, and then reintegrating the data, I think, is the hardest job, and that's what we have to train the most for, which is basically be able to know that electrophysiology, calcium imaging, fuzz, they're always talking about kind of the same phenomenon, but it's first of all, the paradigms are sufficiently different that you're not measuring all of them at the same time, and so you have to create, make your lens specific to the kind of question that you may want answered, and so it's important to value all methods, right? So fMRI, EEG, fuzz. I mean, everybody has their perks and their shortcomings, and we have to think about them neutrally, so that we can give priority to the scientific question and hypothesis, and then we will know what's the right method to go with, to answer that particular question.
Exactly. Yeah.
From a philosophical perspective. Yeah.
Can I ask another question?
Okay. One more question for the session, and then we finish it up.
Sure.
I don't know if you maybe already told us, or if you're gonna see it on Tuesday in the lab visit, but I remember that the EEG was with the cap and with the strings attached. But how do you do that with animals? Because you cannot attach a cap to an animal.
Well, you can basically place them individually. you could attach a can. So I mean, people do EEG recording with non-human primates, and it's very similar actually to EEG with the human. But you can also then actually attach them individually at certain spots on an animal, for instance, because the principle is the same. You just need an electrical contact with the skull, and then you're collecting signal that passes through. Of course, with a mouse, you often could just also remove the skull and place the electrodes directly on the brain for the ECOG. It just depends on the level of detail that you need for that particular question. And therefore, you typically go for the least invasive technique possible in order to answer the question. So that's what the decision that you'll end up making in that case.
Josh, I'm now curious. Did you talk about this the scale? So EEG, ECOG, and LFP and extracellular. You did, right?
Yeah. So not LFP necessarily. So it's
so
more like these are the types of measurement techniques you would use.
True.
But then LFP would be like what you would get from ECOG, for instance.
Yeah, that's right. Yeah.
That's the local field potential. And then
because I might be bringing some of that back in a later class, and I was just trying to remember whether you had mentioned it, but indeed, like it's the same type of signal as the ECOG, electrocorticogram.
Exactly. So then, on the left here, of course, is the actual look of the electrode that you would use, or the monitoring system. So I think this is supposed to symbolize EEG, maybe? This is a weird
It's an
image
EEG
here.
cap, a very modern EEG cap.
Yeah. And then this is, of course, the level, quote unquote, of the way the resolution of the brain. So whole brain versus groups of neurons versus individual connections of neurons versus even individual channels on individual neurons. So this is kind of the scale up and down of the spatial resolution that these techniques
almost makes me
offer.
feel like sketching the type of data that comes from each of these levels on the side.
That was
But
true.
a lot of it is
Nice
just
to add
basically
to.
some wiggling line. So like, it's not exactly.
Yes, exactly. Yeah, there's quite a lot of these things where you can see the data that comes out, and it can just look like gibberish unless you know how to read it and how to make meaningful sense of it. So like if I show like this, I don't think this is a beautiful recording actually. But I think anyone else would be like, oh, what? I don't know what that is. But I mean, the amount of work that goes into getting this quite right makes you love it. That's basic psychology. That more work it takes to make the reward greater.
So I want to share. I still remember the file name of a recording I have from Purkinje cells from 12 years ago, just because it was that beautiful.
Yeah. Exactly.
Sometimes
sometimes you keep them, you keep that image on your computer even if you move different computers. You have to value these things. But
very cool. So guys, I hope that you've enjoyed the discussion. I think that this was a great primer for the lab from the lab visit. It's great that you were here live. And you will probably have to help some of your friends with the knowledge that you've now recently acquired. And I'm going to make sure that everybody watches this before the next the lab visits themselves. Shall we write some instructions, Josh, like so that I post on Canvas for them?
Yeah, so I'm going to make a little map of the floor that will show you which rooms to go to, and then everything that I showed you today that's in this presentation, we can post this presentation in the Canvas as well.
I'll do that now.
These are the sorts of things, if you want to do a little pre-homework of these techniques, these are the things that I've said today, and the things that are on these slides are the sorts of keywords that you might search for in order to start looking more into these techniques first, right?
Brainscape should have the flashcards for most of these techniques we talked about. If you notice that there is a technique that is here that is not in the Brainscape, you should let me know. In fact, I was planning to make the festival assignment for next week, lab visit based, in which you will then produce flashcards for Brainscape that are not the ones we already have. So that's an interesting challenge, I thought. But we shall see how that plays out. All right. So like I said, then the lab visit will be announced. The more details will come as we produce them. And I have nothing further to add. Thank you, Josh. I think that this was a great session. Will serve us for the next years as well. So we can also see how we're aging over the years.
Yeah, exactly. But I look forward to seeing all of you on Tuesday, and talk to you soon.
Have a great time.
Bye.
Thank you.